So one of the technology that we are been shown to be promising is the use of the black soldier fly, and that's because it has been shown it can produce valuable products, that is the treatment residue and the biomass, the larvae. And why? This is because they accumulate nutrients from the waste in the treatment process, whereby the larvae feed different kinds of waste substrates, and then the larvae accumulate it in their body, and those nutrients like protein, carbohydrates, and other compounds can also be used as animal feeds in the production of animal feeds. And then the treatment residue also has some nutrients like nitrogen and protein that remain, so it can be used as fertilizer. So they actually can be used in the production of food. And as I've said, it can feed different type of waste, and then so the larvae grow, and then they go process, they develop into different stages until when they're flies. And this fly exists in different parts of the world, especially in the tropical and subtropical countries. It's a non-vector and non-pest fly because it doesn't feed, so it doesn't have to jump from one food to another of people. So it's considered as a good fly. One of the challenge we have with this treatment is, even though the fly larvae can consume different type of waste, there are some type of waste whose nutrition composition don't really support the growth of the larvae. For example, if we looked into mixed food waste and fruit peels, I've used banana peel and orange peels, you see that biomass conversion efficiency, this is how much of the nutrients of the waste has been converted into the biomass, the larvae body, is very low in the fruit waste compared to the mixed food waste. And this is because of the composition of the mixed food waste. The fruit peels, one of the limiting factor is the protein content. They have very little protein content. And on top of that, it has been shown also the ratio of the available protein and digestible carbohydrates, it should be around 1:1. That's the optimal. So, but then for the banana peel and orange peels, they have a lot of fiber. So it's the carbohydrate that is not available. So we have to consider this type of waste because, as we said, this process, in order to be effective, it has to produce valuable products, and that means you have sufficient biomass, because it has been shown in as another study that the main revenue actually comes from the biomass of this treatment. So if you look into, for example, treating 1,000 tons of waste, you see you get less biomass from the banana peel and orange peel compared to mixed food waste. And if you use it to estimate the value in terms of protein content, because if you use it into animal feed mostly for the protein content, in relation to fish meal, you see you obtain a really less value from the fruit peel. So considering we have this type of waste, we have to look into it. So that's where my project comes in. So in this project, we have looked into develop and assess and compare different pretreatments and substrate blending to improve the process efficiency in the bicefal composting of fruit waste. And also, we assess the availability and suitability of food industry waste for bicefal composting in the low and middle income country settings. And the research questions were, how can we improve the conversion efficiency of the fruit peels into the bicefal biomass? And then what opportunities are available in the current food industry waste management systems in Tanzania for bicefal composting? And for specific objective, we evaluated different types of treatments for the first study and the second study, that is microbial, heat, chemical, and enzyme, and combination of that, to see if they can improve the nutrient composition of the substrate, but also the process efficiency. And then in the second, we also looked at blending of fruit waste together but also with the protein-rich substrate, that is the fish waste, to see if that could improve the protein special composition of the waste and the nutrients in general, but also the process efficiency. And then in the last study, we looked at assessing the availability, suitability of selected food industry waste in Tanzania for potential implementation of the black soldier composting technology. In this study, it was done through papers, and for 2 papers were under pretreatments, 1 paper substrate blending, and the last study also 1 paper. I'll start to present the last paper, where here we went to look, because literature says there is a lot of waste, but is there waste? What kind of waste is there? So this is the first part we went, and Tanzania was a study case. So we went to 3 cities. These are the most urbanized ones. That is Mwanza, Dodoma, and Dar es Salaam. They're also the most populated. And we got the list of food industry companies from the Ministry of Industry, Trade, and Investments, where it had all companies in the whole countries. And we visited all countries in Mwanza, Dodoma, and Dar es Salaam. And we used questionnaire to actually find out their management and production of the waste. So for the companies that had waste, they had brewery waste, sunflower press cake, banana peels, mango seed, coffee husk, and fish waste. And the waste were being produced in different amounts, from less than 10,000 kilograms per year to more than 1 million per year. We asked them if the waste was being utilized in any way. In most parts, it's only the brewery waste and the sunflower press cake that was being utilized. For the brewery waste, they utilize around 25% or less in their own, within the industry. But the other part had, it was utilized by other people. That means they had markets for the waste already. But most of the other food industry waste, most of it was either given away, and if there was no one who was interested, then they just threw it away in landfills mainly. And then we asked, how happy are they with their current waste management? All of them said they are really happy. And we asked if there was somebody interested in your waste, are you willing to give it to them? In case they want to make value to them, they said yes. And because probably they saw that most of them said also the waste had value, they recognized that. So in this part of the study, we see that there's large amounts of unutilized bio-waste, and also some of it, which was not being used, was being landfilled, and companies were willing to let other people manage their waste, and there are also opportunities available for the disciple composting. And then we looked into different pretreatments, and in this part, we chose the fruit waste and the fish waste that were available in Tanzania to look into them and see how we can improve the nutrient composition. And we first looked at them themselves. And for the banana peels, we had one from Tanzania, which is used to produce juice. And the one here that we eat, the dessert banana. And then orange peel. But we can see the nutrition composition differed, for example, for the banana and banana. Different banana, but even for the dessert peel, because we used different batches of banana peel over the years that have been studied, they also had a variation in the nutrition composition. But in the end, all of them produced less biomass conversion efficiency, so the nutrients were not sufficient to actually for the larvae to grow. For the fish waste, we used it in only one study, but all the black soldier fly larvae died, mainly because of the fat content and also it was sticky, so they could not survive because of less aeration, I think. So we went ahead and did the pretreatments, and for the pretreatments, which were done in paper 1 and paper 2, some of them aim to degrade the fibers to make the nutrients, carbohydrates more available, that is the cellulose and cellulose. And then we also applied, we added ammonia, nitrogen, to see if we can add nitrogen for the synthesis of amino acid for the growth of the larvae, and see if it could balance the carbon-nitrogen ratio. Here we had different experiments. That is the microbial, non-protein nitrogen, heat, and enzyme. And for the fungi, microbial, we had 7 days, 21 days, and 14 days retention time. And then for the ammonia, we also had different time and concentration. And for the heat, we also tried a higher heat at 120 and then a lower heat. And also for the enzyme, we had the direct one, and then also at elevated temperature. But also, that is just addition of enzymes, but also we tried to see how, if only mixing of the fungi could actually improve the amount of enzyme and also act itself as an enzyme sufficient to produce enough enzyme for the help with the degradation. Again, we did for the banana peel, orange peel, and mix, and different experiments. And so for this study, we had banana peels and orange peels, whereby we received them, we homogenize them using a blender to increase the surface area, and then we subjected them to different pretreatment conditions. After that, we took them for composting process, and if we did treat for triplicates, then we also did the composting process in triplicates. In paper 1, for the first study, it was more to look into how well the larvae grow into the substrates. So the composting time went for 30 days, and until all the larvae turned into pupa, to see if they actually grow up to that stage. And then so we had 200 larvae, and then we fed every 2 to 3 days. But then for the second study, it was evaluation of the process, and here it was not optimized. So only 10%, when only 10% of the larvae turned into pupa, we stopped the experiment. And here we only fed the first 2 weeks for 3 days. And we did the analysis to see the different modifications that had been done by the pretreatments. And we saw that here I'll only present the ones that really did well to improve the efficiency. And here we saw that all the microbial addition of ammonia and enzyme, there was degradation of fibers. There's a decrease. And then there was only a slight decrease in the enzyme, which was not so significant, but it mainly they also carbohydrate decreased. And the nitrogen was mainly increased greatly in pretreatments where we added nitrogen. And overall, it's only the non-protein nitrogen and enzyme where the CN ratio was decreased. And here it increased. And then for the results, we saw that when treating the microbial, 14 days retention time is the one that produced highest biomass conversion efficiency. And also, when using non-protein nitrogen, the one whereby we mix the non-protein nitrogen and fungi is the one that gave us a better biomass conversion efficiency. And for the second study, we saw that for the biomass conversion efficiency, when you use orange peel, banana peel, and mixture of banana peel and orange peel, it improved the biomass conversion efficiency, even though for the orange peel, it was not significant, but it improved a little bit. But then for the material reduction, it only increased only in the enzyme, the direct enzyme. And pretreatments that didn't work, it was heat at elevated temperature at 1220 and heat at 50, enzyme direct with the orange peel, and then all enzyme and heat treatments, but also all fungi mixture. There was no improvement. And one of the reasons there was concentration of fibers increased, but also most of these had low pH of around 4, so I think it also has a negative effect. And also, even though in some of them, the carbohydrate increased, they didn't improve, so it was probably the type of carbohydrate that increased were not also still available. When forms maybe for receptor is not available. So for this study, we saw the biomass conversion efficiency was positively impacted by nitrogen increase, it decrease in the fiber, and also a decrease in the carbon-nitrogen ratio for the ammonia treatment. And then the ones that really impacted negatively was low substrate pH, increase in fiber concentration, phenols, and tannins. And then when we compared the enzyme treatments, whereby we added direct enzyme and the one we use fungi for the mixture, we saw that for the biochemical, here is the direct, we saw that there was more biomass conversion efficiency, which was really great, but then for the biological, it was not. And then when you come, all this is respiration due to fungi. And here also, this is during BCFL composting. This is the VS loss. But then also during pre-treatment, it had a lot of VS loss. So in biological, the fungi treatment, the VS loss was really, we had a high material reduction in terms of volatile solids, but then it was mainly due to the decrease due to respiration and due to fungi and the larvae themselves, but not really, it was not really converted into the larval biomass. So we thought for the biochemical, it's good for the production of protein, the biomass, but then for the biological, it's really good for just reduction of material. And also because it dries the substrate, because it was really dry, we thought it was good also for mixing with the wet substrate to dry. So here we saw that pre-treatments can actually improve biomass conversion efficiency, and the ones that were good were microbial 14 days, 7 days, a mixture of ammonia and microbial, but also direct enzymes. And then for all the orange pre-treatments, it didn't really work. We think there was some inhibition. And then also for the heat pre-treatments. And overall, fungi also pre-treatments didn't really improve the biomass conversion efficiency, but then the total material reduction was improved. And then for the third paper, whereby we did substrate blending, here we mixed fish waste and banana peels and orange peels. The fish waste was in 10, 25, 50, and 75 inclusion, but then it was varying for the orange peel and banana peel. And here we had 50 mixtures, and also we used banana peel and orange peel. And then we had fish waste, we used the inside of them. And for this study, it was also optimized. So the larvae only went up to the until 10%, turned into pre-pupae. That's when we ended the experiment. And we also had 700 larvae, and we fed from the 3 times during the beginning. And for the results, we saw that the more we increase the fish waste, as you can see here by the colors, the more we improve the protein carbohydrate ratio, but also improve the biomass conversion efficiency. And the highest biomass conversion efficiency was with 75 fish waste. But then with also 75, we also got a biomass conversion efficiency of around 13. So it varied a lot. And this was mainly due to the composition of the fish waste. The fat was more in different type of fish, and then we had small fish and big fishes. Others had more eggs, so we think that also could have impacted. But also, with the 50 and 25, we saw that it was the biomass conversion ratio was almost the same. And this was interesting because for the 50, the protein carbohydrate ratio was 1, which is said in literature that is really an optimal one. And then it was almost similar to the one with 20 fish, whereby it was 0.4. So this was good. And then when you just mix food waste alone, there was not much improvement, especially in comparison with the banana peel. But with the orange peel, we saw the more you added the banana peel in the mixtures, it improved the biomass conversion efficiency. And we thought this was diluting the toxicity in the orange peels. So with this study, we concluded that if you mix fruit waste with fish waste, it improves the nutrient composition and also the process efficiency. And high inclusion of fish waste resulted in variation in biomass conversion efficiency. But then fish waste inclusion we thought was the best for this study. And then for the last study, whereby we looked at suitability for the last part of the fruit waste. So we had two types of criteria. That is the availability of the food industrial waste with accessibility, amount, purchase cost, and competing use. And we also had the physical-chemical criteria. Here, I'll just put a summary. For this, we saw that when you evaluate this type of waste that we had for their suitability as feedstock in BCFL composting, most of them had limiting parameters, which are indicated by red. Either the nutrition content was very low, or it was not as available because it already has market for the case of brewery waste. So in the end, we concluded that each waste alone, it could probably not be sufficient to use as a feedstock because of these limitations. But then if you mix, for example, banana peel and fish waste, banana peel has less protein, with fish waste with more protein, and also as we have seen in the other study that we did before, it could actually work and improve the efficiency and also the nutrition composition. Also, for the banana peel with brewery waste, which we saw was not available, because you need just less of the protein content, it could also improve it. So for this part of the study, we say together with substrate blending, you can actually use this type of feedstock for in this type of composting. And then for the last study, we looked into the implementation, if we could actually apply these results. And then we took the study, for example, for the first study, you see that after 30 days, you get the larvae, the biomass conversion of around 7, but then with the pre-treatment, you actually double the larvae because the biomass conversion efficiency is around 15. But then, for example, the treatment with ammonia and followed by fungi, you have all these extra resources, that is the ammonium, the days, and the fungi, which you have to consider in the end and weigh because it's extra inputs and weigh against what you get in the end. And then we had also other parameter, for example, residue moisture. It has been shown that residue moisture affects the process efficiency, but also the separation. We saw that when it was too wet, for example, for the non-protein nitrogen, it was too sticky and time-consuming to actually separate. So this has impacts on the process itself, how you process the waste, the quality of the residue, and might need more energy, so you need to evaluate this against what you get in the input in the biomass. But overall, we saw that these pretreatments have ability to improve the value we get from the waste, but we have to consider other resources that we put into. For example, for the enzyme, which was the best, it was not dry. And then enzymes have also been shown, for example, in other studies and technologies, that it's really expensive. So you have to weigh against that also. And then for the microbial, which is second best in this case, in terms of biomass conversion efficiency, I've put this controllable because we added water, but then for the fungi, it's really dry. So we think it can actually control the moisture content. But then you need 14 days for this type of waste to actually get a higher process efficiency. So in some settings, it might be not, it might be expensive for the space. So you need to evaluate that. So we recommend future studies to look into adjustment of moisture and substrate pH to see how if that would improve the process efficiency from the beginning if you adjust the feedstock, and then to look into understanding of the chemical and biological processes that happen into the pretreatment process. For example, so carbohydrates increase. If you could go maybe what type of carbohydrates increase or what type of fats, what hindered, maybe that would help, or how the microorganisms function. And then we only use one type of enzymes in this type of waste, probably exploring other types of enzymes that would improve it. And also test the pretreatment methods that we did and substrate blending on other type of waste. And lastly, to implement results, because it was done in a small scale, to see if we can get the similar kind of results. Overall, our conclusion is pretreatments can improve the nutrient content and process efficiency. And then when we mix food waste with fish waste, it also improves nutrient content and process efficiency. And we saw food industry waste is an available opportunity in Tanzania. And lastly, we feel biomass management in Sub-Saharan Africa region and similar settings globally would actually be improved with black soldier fly composting treatment technology. Yeah, and that's all I had. Thank you.
Then I leave the word to Doctoral. Yeah, if you want to, wherever you want to sit.
I would like to stand up. So, thank you very much. Thank you very much for this splendid presentation. And I have to say that I really enjoyed reading your report. It's very well written. So the beginning is good. And I think in Tintin says, I think Captain Haddock said, if the beginning is good, everything will be good. So I've been asked to be your opponent. And the idea is that I will start giving a presentation on composting. And then we'll have a talk about your work. And don't be nervous. Normally I'm a pretty nice person, but of course, I think it's not. I think you'll not be pleased if I just please you. So I also have to ask you questions where I think here maybe she's not really going in depth with the processes or the outcome of the work. So and where you may not be able to answer. But remember that you have just finished a PhD study, and I've been in the field for 40 years, so I know how to mingle out of a situation I don't like, and I think you have not been trained too much in it. So I'm just saying, I really hope you will relax, and I am sure we'll have a very nice talk, and I will learn a lot from talking with you, and I hope the audience as well. So with this, never a few words from me. I will start with a short presentation, and then I will have I've transferred my questions to the PowerPoint, so we'll go through it on that one. So let's see if I can start, or maybe you can help me. Yeah, I think it's better if you start my PowerPoint. Great. And higher front here. So maybe I should introduce myself. My name is Sven Sommer. At the moment, I am a professor at Aarhus University in an engineering institute. And my research area is I have to say that is how to reduce pollution from managing animal manure. And I have to say that animal manure is probably one of the biggest bio-waste resources we have. And that might be also why you invited me here. And I think I'm also I have to speak slower. I'm also invited because maybe your work is how to really to improve the use of these biomasses. And my work has to be reduce pollution from it. So I have a different angle maybe, and I'm going to present that first before we start to talk about your work. So it's about your PhD here, and the title is there. It's already been presented, and I've been very interested in composting. As you can see, this is a big heap of animal manure where you can see there's a lot of composting taking place in it, and there's a lot of losses of carbon, and there's emissions of methane. It's a greenhouse gas and N2O and ammonia. So we have to do something about it, and that's what you have done, Alicia. So it's also why do we compost, or at least why do we compost animal manure or organic waste? It is to avoid that we distribute diseases. That's one very important reason. The other one is to make it more transportable, you could say that, and hygienic too, and stabilize it, because you can see the heat before it will start to compost if you just put it into a sack. And therefore, it's a stabilization and a hygienization. That's one reason for why a lot of people compost. Now, what's happening during composting? It's a really marvelous process, not larvae or wormy composting, but bacterial composting, because you can see you get the temperature up to 70 degrees within the heap of the center of the heap just by microorganisms. And I think, you know, it's just to see that can happen. I think every time I find it so interesting in itself. And that heating is what kills the bacteria. And Björn has developed models that you have to stir it so you can be sure that all parts of the organic matter is heated up to 50 degrees. So your work in your earlier life was to make sure it's hygienic to use the compost. But you can see here what we don't like too much is that you have this huge loss of carbon and also of nitrogen, which we should try to avoid. And then just a little bit about composting, not vermicomposting, but we could call it thermophilic composting, maybe. We talked about it, Björn and I, how are we going to define composting? You can see you have a surface that is cold, then you have a very zone and there's a lot of oxygen diffusing into the heat. Then you have a region where you have a mixture of oxygen. There's enough oxygen, but it starts to become anaerobic, and then in the bottom it's anaerobic. This really, I think if you go from one heat to another one, it will look very, very different. But we did a quite interesting study. We put a plate into the center of a heap when we build it up. Then we could take the manure from half of it very fast and make a photography of the temperatures. And we got something like this, and you can see the surface, which is cold. And then there's an area here where it's very warm, more than 50 to 70 degrees Celsius. And then the interior where it is anaerobic, it's cold again, or colder. And that's maybe you can see here when you have higher temperatures, you have methane production. And where you have the low temperature, you can have N2O or nitrification and denitrification, and N2O emission. And that's what you can see a picture of here. You can see temperatures. You have days from start of composting, I should explain it. And then you have the temperature up here, and then you can see temperature goes up, and then it slowly declines. Everybody knows about this, but I think over here it's quite a nice picture of what is happening. When the temperature is high, you have the methane emissions. Methane is producing at high temperatures. I think we know it, but there's no nitrification at high temperatures. So when the temperature drops, in this case, we had, it perfectly started when the temperature was dropping. And you had the N2O emissions. That's two greenhouse gases, very potent greenhouse gases. difference how you calculate how much CO2 effect it had, but more or less it's like 30. 1 kilo of methane is like 30 kilos of CO2, and 1 kilo of N2O is, I think now it's 270. It changes with time, so it's you have. I think if you're working in this area, I would advise you to look into the reports of the International Panel on Climate Change, because every year or every second year they change these numbers. And then we have the ammonia emissions, and you can see when you have the high temperature, you have ammonia emissions, and then all the ammonia is emitted, or it is transferred by your, in this case, not your larvae, but by microorganisms and fungi into organic matter, so it stops. Now, how can we reduce this emission? We try to reduce it by compaction. So if you compact it, you have no temperature increase, and then you have no ammonia emissions. But when the temperature goes up, if you look at Pardo et al., they were collecting a lot of data from different studies. When you look at that, you can see there's an increase in ammonia emissions. And then they had it where they had airflow through the system, and where they tried to reduce the airflow, and when it was untreated, and I think it's very difficult to stop composting. So it's very difficult to stop the temperature increase. And that's, you could say, that's what I like about it, actually. But we have the ammonia emissions. And if we try to reduce ammonia emissions by compacting the heap and reduce temperature increase, that is increasing the density, then you can see the N2O or nitrous oxide emission, it is actually increasing. So when you try to reduce ammonia emission, you can increase N2O emission. People call it pollution swapping. And you can see actually the same is happening with methane emission. So you can reduce ammonia emission, and this is, I'm sorry I didn't put the name on it, it's one of your colleagues from here at SLU, where they had different covers on the heap. And you can see, for example, peat will, if you cover a solid manure heap with a peat, then peat is acid, and then that will reduce emissions to some extent. Then you can compact, you reduce emissions to by 20%. This is a reduction of the emission, and then if there's a wall around it, then less air can go into the heap, and you reduce composting, you also reduce emissions. Then you can have covers that reduce air inflow and reduce composting and reduce temperature, and that can reduce emissions in average with 50%. The risk is to increase greenhouse gas emission. And then there are the leaching risk. Phosphorus are not leached. Phosphorus is in crystals, or it's absorbed, or it's organic phosphorus components. So they more or less do not leach out of a heap of manure that is composting. But you can have potassium. Potassium is dissolved in the liquid phase. So you have up to 10% of the potassium in the organic in the manure deglitter that is lost this way. But I think what we should think about, and what is extremely important, is that we have to look at what's the efficiency of compost as a fertilizer, in this case of a nitrogen fertilizer. And you can see of the compost, it's not high, not the first year effect, because the nitrogen is transformed into organic nitrogen, to proteins that is not mineralized the first year when the plants need it. Maybe with time you have an accumulation in soil, and then you have residual effects. So if it's not composted, the effect is higher, but you still have a lot of organic nitrogen. So the efficiency means that 1 kilo, sorry, I should maybe start that 1 kilo of nitrogen in the manure has the same effect as 0.2 kilo of mineral fertilizer. So you have to add 1 kilo of compost nitrogen to get the same effect as 0.2 kilos of mineral fertilizer. So it's not a high. It's better with liquid soil manure, and mineral fertilizer, it's 1. And I think that's why every time I go around, I'm more and more focusing on this, on these numbers, because when you are going to talk with a farmer, she or he, they would like to know what is the fertilizer effect of this material. Because if they add too little available nitrogen, they will lose money. and that's a big risk for them. So they like to know these numbers, and you have to be very trustworthy, or you will lose them. They will not listen to you afterwards. So in summary, I think this is what's happening when you're composting. You reduce pathogens, you stabilize the biowaste. It is at the cost of carbon and nitrogen. You could add straw that adds carbon to it, but that will immobilize the nitrogen, so you have less loss of nitrogen in this case, and compaction and covering reduce losses. So that's about emissions, and then I think the two of us, is going to have a talk about your work of your PhD thesis, this pre-treatments and substrate blending used to improve the biochemical conversion efficiency of black soda larvae, treating all kinds of wastes. So I don't know if we should sit up here, have two chairs, or if you prefer to stand.
No.
So let's see how. So maybe I was just, when I read it, then it is called vermicomposting. And you can see my thinking of composting is maybe a little bit different, or maybe not. So I would like to ask you to kind of definition of this vermicomposting, why it's called composting.
Black soda fly composting.
Yes.
We define it as degradation of the organic matter using the larvae at anaerobic conditions.
So it's aerobic conditions, and it's transformation of organic matter.
Yes.
Yes. I'm just going to show you, when I looked into, I think, Wikipedia, I looked into then it's like, we have this aerobic, at least we agree on that. Then it's a microbial community, you are saying black soda larvae?
Yes, it's the black soda fly, but then it works also with the microbial community, because also inside it and also in the waste there is microbes. So it's both of them working together to actually degrade the organic matter.
So if you take the
enzymes.
Yeah, sorry.
Yes, and also they produce also the enzymes.
Yeah. So when you take some organic waste, and you have black soda larvae, what's the process from organic waste into larvae protein? Do you understand my question? Or?
So they break down the organic matter.
But who do it?
The black soda fly itself ingests the food by swapping it, and also it produces the enzymes. And then with also the help of the microbial community from the larvae itself and the ones that are there, they degrade the food in different sections of the larvae, inside the larvae, until it gets out, like the remaining, that is the treatment residue.
So do you think it's mostly the black soda fly that takes up the organic matter, or do you think it's microorganisms that is taking up, and then the larvae eat the bacterial or fungi? Right?
It's microbial.
Yes.
That's one that really degrades it, and then the larvae uses it.
Yeah.
Yeah.
So the larvae take up actually the microorganisms.
Yeah.
So it's more or less, that's also driven microbially larvae, microorganisms. So, and it's, you're producing a stable substrate. The only difference is the heat production. In a way.
You have higher temperatures.
Yeah.
That are really high, and as they increase the temperature, but not more than, if it's 20, not more than 3 degrees, 4 degrees more. The process does not increase, but it doesn't have the flame of fluid.
What would happen actually if composting started and you had 50 degrees, 60 degrees?
Well, we kill some of the microbial community themselves, even the larvae themselves, they will not, maybe they will even escape, or they will not really take it up.
They can move away.
They can move away.
Interesting. So you've convinced me actually, except for the temperature. I love that heartbeat. Then when I was reading it, your report, then you have decided to, to end up with a survey of bio-waste production in Tanzania. And I would like to, why, you can start and you can have it in the middle and you can end with it, but you had this decision. Could you explain me why you would think it's great to have that at the end of your study?
Well, we did a preliminary study to actually see first what if the waste was available, and then we found out that it was available, and then we continued to do, because of the time and the structure, we just structured it in the end to actually go and do the survey now to establish what is there. So the survey came later on, but before that we actually did a preliminary study also that we have not documented.
No.
No.
I'm just wondering now, for example, you can also, today you showed that brewery waste, there's plenty of brewery waste, there's plenty of sunflower oil cake, as cake, but it's already used. I think I remember it was for feed to animals.
Yes.
Do you think that if you had made, for example, a cost analysis of using these two biomasses, if that would have been beneficial, cost beneficial also, that you could have, for the producers of?
For the producers' perspective.
Yes, for the breweries, yeah.
Yeah, I think it's really important for one, before they establish, to actually do a preliminary assessment of availability of the waste itself, and then its composition, to see how good it is for the larvae, and if all the costs involved in the waste before actually going into it, if you really want for it to produce revenues and for the process to be sufficient. So you have to, so for the brewery waste, I think because it's already being used as animal feed, as a direct use, probably it's not really a good stream to look at, because I mean, in the sense of protecting the environment.
But I just wondered, do they sell it?
Yeah, they sell it.
They sell it.
Yeah.
And so my question would be, could you, by producing the larvae, make a higher profit? Could that be an option?
It depends with the how much you sell the larvae themselves.
Yes.
If the value from it is good enough and also produce enough biomass and it's reliable, you can. And depends also the labor and all other costs. Because it's not only the only thing that determines if you can actually get a profit out of it.
But could it have been an of interest? Because I think this, as far as I remember from brewery waste, there's quite a lot of nitrogen in it and carbon, and so it could have been a. Yeah. I assume it is a good.
Yes, it is a
good biomass for larvae production.
Yeah, it's a very good biomass for larvae. Yeah.
But and then I think maybe if you have made if you have included it, then my question is could this have been an alternative to feeding the animals directly? Larvae production, larvae protein production.
Yeah. It could have been an alternative to the larvae production.
So, So, and that's my next question is also a little bit linked to that, because when you're using these peels from oranges, then you have these problems with limonene, and actually we had, and this is an example, and I don't expect that you know, it's because we had students who worked a lot with D-limonene, and it has a high value on the market. So when you're carrying out, now we're looking at this larger scale, when you're producing this these larvae, they will compete with other products that you can make out of the bio-waste. When you are doing these studies, is that part of your risk analysis or cost-benefit analysis to see could it be used for something different?
We have not done that.
No.
I've not done that. I've not done cost analysis. I've not done the study just aimed to actually see what kind of waste is there.
Yes.
How available are they? What is their use? Just to get that information and then to help now.
Yes.
And so I've not really done more into cost-benefit analysis, and if there are other people they use, I just know if they use it or not.
Yes.
I didn't go into details of even how they use it. Not really.
No.
Yeah.
Yeah, I'm just wondering if I think in the years to come, we will have more and more use of all kinds of biological materials, and there'll be a lot of competition for it. At least that's what I hope, that we are not wasting it as much. And then I think it's these cost analysis and alternatives, we have to look into that. So that was just, nice. Yeah. And then then you had a range of treatments, and I just, now we're talking about your review, and then we'll talk a little bit about some of it again, but that's the way I went through it. So how will heating to 120 degrees, that means a little bit of pressure as well, how will that affect your biomass?
One, when you heat it, it breaks the structures, breaks the macromolecules,
yes.
And then it produces less carbohydrates or compounds, which are shorter chains. But for this case, in this type of pills, actually saw that the fibers were actually grading into phenols and tannins. And this we saw it correlated negatively by mass conversion. So they had some toxicity that they were not still available for the larvae, and they were not really grown. So that's how it affected it.
And then microbial pre-treatment, I think it was bacteria and fungi you are using, and so I would also, what are they doing to the biomass?
They're also doing
in the whole treatment chain.
Yeah,
it's very general questions, but
they are degrading also the fibers, and on top of that, we saw they also dry the substrates. Actually, we noticed that.
So why do they dry?
I think there's a lot of respiration, and the water evaporates because they like the simple carbohydrates. The one they break it, so it becomes loose water, and then probably.
Is there some heating as well, or a little bit of heating?
No, we didn't do any type of heating.
No.
But no, we didn't do any.
Not a little bit? Or
no, we didn't. No. We didn't do any kind of. It was just under room temperature.
But didn't you cool it, or?
After.
In your chamber, didn't you have a kind of cooling to keep it below 37 degrees, or?
Yeah, it was just under room temperature, 28.
Yes.
Yeah.
You didn't have to reduce the temperature?
No.
No. Okay.
We didn't reduce any kind of temperature at any point.
And fungi, how will they contribute to make biomass available, lignocellulosic biomasses? What's the benefits of fungi?
They degrade it because the larvae, for example, they can't really digest the fibers themselves to actually, because they need carbohydrates, they need the valuable protein, the digested one, to actually use it, or the fats for energy or for making the body cells, or like for growth in general. So, the fungi is the one that helps in breaking them so that they're actually available, so they can be used for the larvae.
And how are fungi doing this? What are the processes?
They use enzymes mainly.
And which kind of enzymes?
So, for example, for the fibers, we have hemicellulose. So they have hemicellulase, and then there's cellulase, and then if it's lignin, there is lignase, and then like there are different kinds. Protein, there's protease, there's pectinase. Like they have different depending on what kind of fungi also.
So is it enzymes or exoenzymes, or is it inside the fungi or outside, or?
It's inside.
Mhm.
Yeah.
And then you come to the ammonia, which is an old way, also for using straw to cows, for example.
Yeah.
So how is what's ammonia doing to the straw?
So ammonia also, it provides extra nitrogen,
Yes.
and then at the same time it has been shown that it also helps in the deposition of the fibers in
Yes.
itself by use of microorganisms, and also it somehow also helps in the synthesis of the enzyme.
So do you think ammonia is contributing to the bacteria, or is it a chemical process in itself?
Yes, it is. Yes, it is.
A chemical process.
Yes.
So when you add the ammonia, do you think the larvae can take up ammonia and carbon and produce proteins, or do you think it goes through other pathways?
I think it goes through a lot of pathways until you actually get into the amino acids and the like they break them down.
So would you think it's bacteria?
Yes, it is bacteria.
So again, we started to talk about you have the organic matter, you have bacteria, you have larvae.
Yes.
So in this case, what do you think if you put ammonia to your biomass?
Yes.
Then you have what will the if you had N15 or you had added, would they then take ammonia N15 up directly, or would they take bacterial protein up?
If it has been broken, I mean the ammonia itself or no, I think it's the ammonia actually works on it, and then they take up the product and the microorganisms themselves into their body, and then they use it. Yes.
So if they take
not
up
ammonia directly.
Sorry.
Not ammonia, like the products of
of proteins.
Yes.
Of bacteria
Yeah.
or fungi.
Yeah.
Then I was wondering, if you're in the gut of these fly larvae. In the beginning, I think it was pH 6.2, and at the end of them, where they excrete, it's 8.5.
Yeah.
And you're saying that if you just have the enzymes and you add it on the day, you also add the larvae, you have little effect.
Yes.
Am I right?
Yeah. If you have the enzyme.
Yes.
And then you add the larvae. That's what you said.
Yes.
You add the material and the enzyme, and then you add the larvae.
Yeah, on day 0.
Yeah. Yes. Yes, I had that also.
Yeah. And they were not too happy. Was that because of the low pH? Or?
No, actually, they were happy when we added the enzyme and the larvae together and the substrate. That's when they were really happy. That's when they grew.
And that was because?
We don't really know yet exactly how the process is, but we think we saw there was degradation of fibers, but also think maybe the enzyme, the larvae themselves, the activities, like the consuming of the food, the movement, helps really to mix the material and the enzyme to cause that reaction and degradation. Maybe it improves it, or probably the enzyme itself, maybe it enters and still works inside the body. So it's just still a speculation. I don't think we know much about it. Yeah.
So could I think of the larvae as a tube where you have a lot of mixing and a lot of bacteria, and where they actually more or less like ants in a heap, make the bacteria produce bacteria protein and then they take it up, or
no,
can I look at it that way, or?
No, they also produce enzymes themselves, because their mouth has enzymes, I said before, which also helps the degradation.
Yes.
And also they're parts of the of the body also have enzymes which
Yes.
also degrade. So what enters is actually being degraded by the enzymes which they produce, but also the microbial community, which is also in different parts.
Yeah.
So it's a combination
Of everything.
yeah.
Yeah.
I think it's a combination from the beginning.
Yes.
Yeah.
So do you think, one of the worries we have with bio-waste are these pharmaceuticals, for example, if you use sewage or organic contaminants, that can be in all of the waste that you present, orange peels or banana, I don't know, peels. I don't know if you spray them with fungicides or banana. I don't know anything about banana production. Can this process, vermicomposting, reduce that?
The business for composting. Yes, it has been shown that the larvae are able to, the system in them is able to degrade some of the pesticides and break them completely. And but then I'm not sure if it's all of them. Some of them are being shown to. So probably there is need to see what kind of pesticides they use and see if it's being degraded.
Yes.
But some of it has been shown to actually be degraded completely.
So do you think do you think that there's a risk that this will be a pathway of contaminants, not heavy metals, because heavy metals you cannot really get rid of it, but of these organic contaminants, do you think that the larvae will take it up and it will be a pathway of these pesticides or pharmaceuticals to go into your animal, because the larvae is going to be feed and then be transported up in the feed chain?
Yeah, there is that risk.
Yes.
There is that risk in some point. Though it has been shown they actually can degrade some of them.
Yes.
But I'm not sure if it's all of them.
No,
so there's always a risk.
Yes.
I think there's need to keep on doing research and determining each different kinds and see if they can degrade all of them or some of them.
Yeah.
Yeah.
I assume it's the risk in all kind of when we want to use organic waste.
Sure.
And then I have a lot of text here. It's the different treatments you have. Now you say when you're using the fungi, you have a discussion on when it works best, and maybe you have already said that you have this 7, 14, and 21 days of treatment. So in brief, you have said that it's why is it these 14 days? Why is it not 7 days or 21 days? Or and will that depend? Maybe the additional question is, will that differ between different bio-wastes?
I think it will differ. It depends with the type of substrates, like the composition. For this type, for the banana peels, we saw that. I think maybe 7 days could have been short. The degradation happened for, but not to the extent whereby there was available nutrients, like which are digestible and utilized. And for 21 days, for this type, we saw, but probably thought the fungi actually consumed it. So that's why we think maybe 14 was optimal. But it might differ with another type of substrate, maybe with less fibers or more fibers, and what type of fibers or carbohydrates that are actually in there.
Yes.
Yeah.
So this juice peel, banana peel, what would you think? As far as I remember, you said there are more fibers in that one.
Yes.
So what would you think? Would it be 14 days, 30 days, 21?
It's hard to say for now, but because I don't know what kind of fibers actually that exists in there. We didn't really go into the detailed chemical analysis. Probably need that now to understand. Does that mean if it's more fibers, it's less time, or it's just the type of fibers also? I think
but in general,
another study.
but
it's
if you,
easy to assume that.
to assume.
But it's easy to assume maybe, but also it depends also with the amount of microorganisms and support also, because that's also determines probably, maybe, I'm not sure. I'm
No.
not sure if we put it, you need more days or less days. Maybe the same.
Would you think less days would be? Would you expect fewer days or?
No, I
No.
wouldn't expect
No.
fewer days because it's still banana peel.
Yeah.
Yeah, but I'm not sure if I can expect more
No.
days. But probably it might be fewer days also, maybe 10 days. So I
Because,
don't know.
okay, it depends on the composition of the
Yeah, it
fibers.
depends on the composition of the
Yes.
fibers. I'm not sure. Yeah.
Then it's non-protein, it's ammonia. And yeah, I think we already talked about what's the reaction. So if you only had ammonia, was that? I was just wondering if you apply ammonia and if you have no pre-treatment time, 0 days, would you think that could work like the enzymes?
We did that. We did that, but with heating. We saw it didn't really, but then we didn't test it alone, we still tested with fungus. But it didn't really improve, but we still saw some degradation.
Do you think it's ammonia? I think ammonia is lethal to a lot of organisms. What about the black soda fly if you have a high ammonia concentration?
Well, if you're very high, I'm not sure if it will actually affect them, but maybe because it's really toxic in most of even just for us ourselves, you really feel bad. But so I think it might affect them also in one or another.
Yes.
Yeah, I think so.
And then this, you had this heating to 120 degrees, and I was just wondering, if you have the biogas expert, they would say steam explosion, having heating and then get off the pressure immediately.
Yeah.
Were you thinking about processes like that, or?
Just heating.
Steam explosion, you heat up and then you release the pressure very fast. And you have added some also, oxygen.
No, not really.
No. Then we've talked about this combination of chemical and microbial. Now I'm wondering if in Tanzania and also in Sweden, Denmark, I assume these black soldier fly vermiculture, the people who are going to manage it would like a simple treatment and very, very cheap. Yeah. So of all the ones you have mentioned, what right now, what were you thinking when you were, talking about enzymes? Would that be feasible in the future? When you started this, would you think that people could get that so in a way that the production is cost efficient? Yeah, I think being engineered.
I think if it works, there are people who might have the money to actually do it or to actually buy it, but also there are technologies probably that could actually produce it, find a way.
Yes.
So I think there is a possibility, though it may not be very for everyone, but you never know.
No, I'm just wondering if the market for the protein will pay for it.
Yes. If the market for yes.
So it is. You can see this market, or?
Yes, I think there is market for both of the products because we need a fertilizer.
Yes.
Because it's an agricultural countries whereby we need fertilizers, and so far there is need for protein sources. That means if you have this protein source and it's high quality and more available and reliable, definitely they'll go for it and you get market out of it. And then at the same, so the same as for the protein, but also the fertilizer, both of them, like in the food chain, we need both for animal feeds and also the. So I think there is market.
Yes.
Yeah.
Then I go to your study. So you have kept the temperature constant at 28 degrees, you're saying? So that's what so, but in paper 1, you had 0.8 larvae per square centimeters, and the next ones you have 2.9 larvae per square centimeter, sorry. I'm just wondering, would that make a difference in your results?
Yeah, it could. Because probably it would, even though the depth was within, but then larva density, we gave them more food, but then they grow. Here, for the first study, they just had much. There was extra probably because it was more, but for the second one, it was more optimized. So in terms of like amount, I think they all got sufficient, so it might not really affect it. But also for the other one, it went for a longer time, and I don't think there is much difference.
No?
No, I don't think there was significant effect into the system. Maybe a little bit.
If the first study that was more without mixing the biomasses, or because I don't remember it. It's the question.
The first study. The first study.
In paper 1.
It was just banana peels.
Yes.
And then we had with different pretreatments.
Yes. And then 0.8.
And then yes, and fewer larvae.
Yes.
And then for the second, it was also still banana peels, and also we had different orange peels, and then we had also another mixture of banana peel and orange peel.
And then you look at this biomass conversion efficiency.
Yes.
I'm just wondering if you have less larvae, then that can be lower because you have less larvae.
That's what you mean.
Yes.
Yes. Because the larvae has the maximum amount they can consume. Yes. in a substrate, like they can't take more than that. Yeah. So that was the question. So they can't take more than that.
No.
Yeah. So I think that is what was available. But if you give them more food, it means there is more available larvae. There is more available. But then also this one for the second study, it was like we gave them the amount that they need.
Yes.
To survive. Yeah. So that means I don't think that's why I said I don't think there is much significant difference like between the two studies. I don't think they can have more than that. Because both of them they got sufficient amount of
yes.
Yeah.
But then I think the BCE couldn't that be different because of that? Because the number of larvae, or
because
if you compare paper 1 to paper 2 and 3.
Yeah, because they had more food.
Yes.
Yeah.
Of too few larvae per cubic meter centimeters of food.
Yeah, maybe. Yeah.
Then a little bit into you're using this. Oh, sorry. You're using this relation protein to carbohydrate. And then you're saying, you can see here to the right, I've just collected from an article the rate conversion factor that is, if you have the nitrogen, then it is said that the standard is 6.25. That's the weight of the proteins. I think you were saying that with fish, it was 4.5 or? something like that, on the
Pardon?
top of my head. Am I
Pardon?
right? You're saying that the ratio between protein to nitrogen is
Okay.
4.5 if it's fish protein.
It's one and two. Yes, one to one. The protein.
No. If you have measured the nitrogen concentration,
Okay.
then you can calculate the protein
Okay. Yeah.
weight or
Yeah.
And I think you are using a ratio of 4.5 conversion.
No, for me I use 6.25.
6.2, but I
Yes.
think you were
But then
discussing.
there is a study that say that this one might not be representative
No,
because it includes some other type of protein like chitin, and so they suggested that ratio 4.
yeah,
Yeah.
do I remember wrongly, or was it? I thought it was quite low, the ratio. Yeah. Because if you go to the right, I think it's quite interesting. We are all using 6.25 now. And I learned it's from casein. I think it's from milk. We have that ratio, and you can see it goes from 4.71 up to more than 6. And the reason why I go into detail with that is this is how it looks, a protein, is that you're using this ratio of, I think maybe it's the next. No, but it's because you're using this ratio of protein to carbohydrates. So you're also calculating the carbohydrate concentration using a factor, which is from the VS, and you multiply with divide by 1.8, and then. So I'm just wondering, why are you not using the N to C ratio instead of protein to carbohydrates? Because
Why don't I use
the nitrogen, we use the CN ratio very much in soil testing, and we all of us know now that it's not really a good number, but you're taking it even further, I think, to the protein related to carbohydrates, where you have these problems in calculating the protein concentration and carbohydrate concentration. Why are you using that ratio instead of the N to C ratio? Do you see mine?
Well, we are using the protein. I don't know much about that. I mean, we have done the CN ratio.
Yes.
I have that, and the protein carbohydrate ratio, just to see how they change, how available they are, and I mean, what's the ratio. But I'm not sure about that one you're talking. I've not used it.
No, but I'm just wondering if you have this, if you mix the fish and it was the banana peels and the oranges.
Orange peels,
Yeah.
yes.
You mix the fish and the orange peels.
Yeah.
And then from the fish you get a lot of nitrogen. And it's calculated as proteins.
Yes.
And then you get, you show very nicely, you had a very nice figure to the end, saying that it's the ratio could be 0.5 instead of 1.
Okay.
Then I'm just wondering, is that also related to these ratios? This way of calculating protein and calculating carbohydrate? Because you have this variation. It will, of course, go in another direction.
Yeah, so for the way I use protein carbohydrate, it's what we give them.
Yes.
And that's why I just checked what the protein and what's the carbohydrate. But then for the CN, for the first study, I use CN, not really protein, because we actually gave them the nitrogen, not the protein. And we're not sure if it was all converting protein in the end. So that's why there was that difference.
Yes. But do you think that when you get this result, that other people say it's like a ratio of 1 to 1, then you're showing it could be 0.5 to 1, which is also fine? You showed that in the last, when you add fish and mix fish and orange peels, then the ratio is quite large. I'm just wondering if it's also related to the way you calculate protein concentration and carbohydrate. Maybe that's not so precise. Or
I don't think it's just a ratio.
Okay.
Yeah.
Yeah.
In the end, it's just a ratio.
Yes.
Yeah. If you use the same method, it will still be a ratio.
Yeah.
Sorry to disturb you, but we may have a break.
Break. Yes.
If
It's okay. There'll be half an hour more, and that's great. Yes.
Thanks a lot. Then 10 minutes. We will start 2:40 exactly.
Yes.
Hopefully.
So yeah, everything is fine now. We have at my institute been working with biogas production, and biogas production we measure volatile solids exactly the same way as you're doing, and we are very concerned when you are drying the biomass first to get the dry matter content. There's a loss. There's some of the there can be some alcohols. There can be some organic acids that are lost.
Yes.
That would really be good for for the bacteria to as a substrate. And and then after you measure the dry matter, then you measure the volatile solids by ashing, as you have described very nicely. So with the volatile solids, I think I have to ask you a little bit. So if you have if you have a lot of, let's say, volatile acids, organic acids in your biomass when you start, it could be brewery waste maybe. Yeah. And then you dry it, and then to get the dry matter, and then you measure the volatile solids by an installation afterwards. What would happen to would your concentration of volatile solids be correct?
By after heating it up.
Yeah, after first you are measuring this dry matter, and then you incinerate the dry matter, and then you calculate your
solids.
There's a dry matter minus ash divided by yeah. So will it be correct, your volatile solid measurements?
I think they would, because we have removed the water and the inorganic part, like the ash, and we have also heated at lower temperatures. So I think it's correct.
Yes. Mm-hmm. So if, for example, there was alcohol in it, also if you had the lower temperature, would you think you would keep the alcohol in your dry matter?
At lower temperatures?
At 60 degrees?
Probably.
Yeah.
It depends with the evaporation rate of the organic compound.
Yeah.
Yeah.
So you have this risk of that your volatile solid is measured too low if you have been thinking about it. I can hear that.
Yeah, it was mentioned before.
Yes.
That the temperature affects the type of organic volatile solids.
Yes. Because I was wondering again, you have this BCE, biochemical conversion efficiency. I'm just wondering if when you are measuring your volatile solids this way, and we know there is a risk, some organic components can be lost during drying and measuring dry matter. Yeah. And if you do that, you reduce your VS. It's incorrect. Could that be a difference between when you are measuring VS of the larvae and VS of your biomass?
Probably, yeah. Because it depends on the composition again of what type of volatile you have in that substrate that you are shading.
And then I was just wondering, would that be if you have a larvae and if you have this mix of organic matter, if that would be, if there's a risk that the volatility of the organic matter are lower or higher in the larvae compared to your biomass?
Yeah, there is always a risk.
Yes.
But I've not really done to see the differences. No. Probably it's something to do, but But I think the type of, unless there's other type of volatile matter that actually evaporate at that temperature, maybe.
Yeah. But if then, for example, you would say that black soldier larvae organic matter is not so volatile, as the biomass, what would happen to your BCE?
If it's not so volatile.
Yes, so it's not lost in your dry matter.
That means you'll have more VS in the larvae and less of the and so it would be greater.
Yeah.
Yeah.
So it will be a kind of artifact.
Yeah. It will be a high biomass conversion.
Yeah, or opposite.
Yes.
Yeah.
Yeah. So that could be an effect.
Yeah.
Yeah.
Yeah, we talked about this, and I think we also talked about this. We talked about the brewery waste. It's something I remember as a child. Actually, we had a brewer in my small village, and they were selling or giving their waste to the pig farmers.
Okay.
So we have talked about this. So I think that's quite interesting to see what's most beneficial for the brewery, for example. Mm-hmm. Then I was wondering, you have focused on some very nice bio-wastes from the food industry, but we also have something which is not so nice, and there's a lot of it. There's household waste, for example, and there's agricultural waste, which is manure, and then the agricultural industrial waste, which you have been using. Yeah. So do you think that you'll be in a position to use the two other wastes, or is it just as good not to use the larvae and apply it on land without, or?
I think all of them can be used.
Yes.
Yeah, maybe it's just all of them can be used because the household is like food waste and all that. It's really a good mixture. There's only a risk of maybe contamination since it's not segregated, but then if you can get it and you're sure it's safety, I think it's really good substrate and it's available also. Also agricultural waste, we have a lot of that from the farms and production animals, which is agriculture. So yeah, it also can be used. So I think all the three wastes can be used depending on their availability. Yes. They all have potential.
So I don't know so much about Tanzania, so I'm sorry, but when you have dairy productions, are your animals on free ranging or are they in the animal houses being fed with? So do you collect manure or not? It's just for curiosity.
Oh yeah, we collect manure.
Yes.
In most cases, we collect manure because they are being raised. In some parts in our country, they just free range. Yes. But then in the cities, it's more in the housing. So they collect manure.
So you have manure collection.
So we have manure collection.
Is that increasing? It's nothing to do with.
Yeah. I'm not sure. No. I've not been really
it's not
following
your area,
it up.
So
yeah. But I know we eat a lot of cows. Yes. Cow meat. And there's a lot of production of it. Some of them are being brought from the countryside, some of them are being actually raised here. So at least I know there is a lot of production, but I'm not sure if it's increasing.
In the housing works, yeah.
I'm not sure about that, yeah.
Then I think I have already. So there's this difference. Why would you choose these dessert peels? Is that because you were in Sweden when you made the study, or is it?
Yeah, because I was here. I mean, originally I wanted to do peels from the juice peel, the banana peel, but then the logistics of transporting it here was not easy. No. So we said, well, because it's just peels, let's just do here. The ones we have here that is more valuable.
Yes, but if you're going to give advices to producers in Tanzania, can you use the experience you have from Sweden, or do you have to carry out a feasibility study first?
I think I would prefer to also do a feasibility study to make sure that you get, because you have seen there's but overall, for all the type of pills, generally, we have low conversion. So I think we'll still need the pretreatments and substrate blending because they all have low protein and a lot of fiber. So for the part of pretreating, definitely we'll need them. But then the only thing is maybe if the condition we applied for the dessert pill will be exactly the same, or they'll need a little bit of modification. That's what I'm not sure. Maybe you could start with that, and then if they don't work, you can see how you can make it better. Yeah.
Yes. And then you're saying dry matter content increase in most treatments. So I assume that's due to water evaporation.
Yes.
Then you have this variation in pH, where the fungi increase the pH a little bit more than in the other treatments. Can you tell me why you have these variations in pH between treatments?
pH, it means because the bisexual composting, the way it happens, when the degradation happens, then it leads to production of organic acids, and then later on it stabilizes. The process goes into production of more of the alkaline compounds. So basically, that's what happens. So basically, fungi increases pH. That's what we saw, that it does. We have more of the alkaline treatment residue. So I don't know, maybe because it also started at a higher residue, substrate pH, but that's something we have to study further.
Yes.
Yeah,
because enzymes did not do that.
No, enzymes did not. I think the fungi is really, it's really like that. Yeah, it just makes the process more stable, probably by having a higher pH.
So then I was looking into the fish. I had to translate it into Danish because, so as far as I can see, then many of these fishes are freshwater fish or brackish water fish. And I think it's the Baltic Sea is a brackish water ocean. Would you think that there's a difference between, if you go to Tanzania, I think it's the Pacific Ocean, it's more salty. I think the salt concentration is higher. So would that affect the composition of the fish?
We might find some of the fish there, but some of them totally different. The type of fish, yes, and I think it might affect a little bit because we also saw here in this study that there was different composition of the fish. We actually experienced it, even the fish in the same, but from the same place. We saw that because we used different types and it brought a lot of variability. So I think, yes, it might affect a little bit.
Yes, yeah, there might, So I'm not sure if freshwater fish can be fed and saltwater fish can be fed. I was just, but the fish you're looking, because you have the Lake Tanganyika or Tanzania.
Yeah, we have.
There are carps maybe there.
Yeah, we have
herbivorous fish.
Yes,
yeah.
we have.
When the ocean.
Yes.
It's interesting. So again, you have actually to make tests. Also with it, because every time, I'm just wondering, you have to set up a consultancy and giving advices every time there's a new biomass, more or less.
Yeah, I think if we use a big percentage of fish, it might really vary. But I think fish generally has a high protein content.
Yes.
So if you use a little bit, like there is soap, probably you might not see a lot of variation, you still improve the process. But when you're using it alone, maybe because for example, in another study that was done by the same research group, for them they use fish as fish, but then they could not go to more than 20%. But for me, I could be able to even increase more of the percentages in the West. I went up to 75
Yes.
and they survived. So I think it depends with the type of fish and its composition, how much fat, what kind of fats they have, for example, and all that.
Yes.
Yeah.
I was wondering if it's the concentration of fat. As far as I read your report, it was the fat percentage.
I think fat also really was high, and we also saw the difference even when we were working with the fish themselves. That some had a lot of fat, some didn't have.
Yes.
So that's why we thought the fish, the fat.
Yeah, we talked about this ratio of 1:1, and you could see with the fish. So I think we will jump over this one. Yes, I was wondering this. So why is this? We'll just look. We have talked about this carbohydrate to protein ratio, or protein to carbohydrate ratio before, so we'll leave it. But why is fat a problem in this production?
Why is
a high fat concentration a problem for the production of the larvae, black soda larvae, fly larvae?
One part is when it's high fat, depending on the type of fat, it doesn't allow aeration to go well. So it's easy to create anaerobic conditions, and then the fish just die because there's no aeration in the system. But another reason could be the type of fat, because it's just fat, but then the type of the fatty acids. For example, maybe others are more toxic, maybe they hinder growth, or they're not available also. So I think it's a combination of all that so far.
Is the biggest problem that there's no air exchange if there's a high fat, or is it the other problems? What is the biggest problem of it?
What do you mean there's no exchange?
Yeah, if you said if you have a high fat concentration, you have no air exchange.
Yeah, that is also one of the big. If it's there, it overshadows all the other reasons. Yeah.
Okay, it's just. And with the carbon peels, I was just wondering, is it because with the orange peels, what's the biggest problem with orange peel?
With orange peels, I think the biggest problem is the phenols and the oil, the D-limonene.
Yes.
Because even when you do even it alone, even with the pre-treatment, we have seen it never really works.
No.
And we think it affects both the larvae themselves, because it has been shown at least with the housefly, and also the microbes.
Yes.
So they all can't really function in the process.
Yes.
So on top of having fibers and less nitrogen, that is also one of the big limiting factors.
When you use this peel, did you wash it before, or?
The peels?
Yes.
No.
I was just wondering. I assume there could be pesticides on orange peels.
Probably. I don't know.
Because
Maybe.
So
why do you assume there could be pesticides? Because you think they're grown with a lot of pesticides?
Yes.
Okay.
That's what
Yeah, maybe.
And then we have talked about this fungi mix, and we have talked a little bit about the ammonia, but why do you think that it works quite well if you have ammonia first added and then you follow with a fungi mix? You have a relatively good BCE.
Because it's a double degradation.
Yes.
Both, and also more nitrogen. for both. So I think it's really good.
Do you think fungi can take up the ammonia? And if ammonia and carbohydrates and produce fungi, organic material? I don't know anything about that.
They degrade it.
Yes.
Yeah. And then, yeah, and then it helps in the synthesis of the product. So in the synthesis of the body structure, for example, for the larvae and other compounds in the body of the larvae.
Yeah, we talked a little bit about this pH increase, so we'll leave that. Okay. And then you had all these effects. I would just like to know a little bit about, you were working on 28 degrees and 50 degrees, and you came up with 24 hours was a perfect treatment time. Could you just explain to me why is that?
For the enzyme.
Yeah, that it is really, that these, that you have this temperature effect on activity, and you have an effect of timing. It shouldn't be too long, it shouldn't be too short.
So for the enzyme treatment, for the direct one,
yes,
we only did with the 28 and 50.
Yes.
And for the 28, is where we saw an improvement in the biomass conversion efficiency, but not for the 50. So your question is, why do I think it improved it in the 28?
Yes.
One, I think still we got degradation of the fibers as we had hoped, and we didn't see much increase in the carbohydrate, maybe because we just pre-treated and did the analysis, like we didn't leave it, so it was hard to see if it increased or. And so I think maybe the activity of the larvae and the enzyme itself inside the larvae, and it helped in the degradation process, and also
so this mix in the larvae.
Yeah. But then for the 51, we saw the pre-treatment dried the material, so I think it concentrated the nutrient content of it, because we saw an increase in all the nutrition composition, that is the fibers, the carbohydrate, the phenols. So even just considering the fibers, I think could have an effect, because they can't digest it, they can't utilize it. But also, even though we saw the enzyme worked, because we saw degradation of, more carbohydrates content, but probably it was not in forms that could be utilized. And we also saw in we didn't do this chemical analysis to check what type of carbohydrates were activated, but at least another previous literature, they say they looked into different carbohydrates, and they saw that some type of carbohydrates, they don't favor the growth of the larvae, like arabidons, I think, and some others. But some glucose they helped. And for them, in that study, they used different type of enzyme, and they also looked at enzyme activity, and also saw that hemicellulose, for example, it hindered it favored the production of these other types. Mm-hmm. And they also use orange peel, for example. And for us, this type of sulfur, we also use hemicellulose. So probably that could have had an impact. So maybe in the end, we think maybe the type of carbohydrates were active. And also phenols was also concentrated, and phenols was also seen that it affects. So these are the reasons.
For it, yes. Yeah. Yes, but you got it at 40 to 60 degrees, and then Lindbergh et al. Got it at 28. Am I right?
Optimal temperature
Yes.
range. Yeah, the literature between 40 and 60, and
It
surprisingly.
depends on the inside.
Yes, surprising as we got it at 28. So
Yes.
that was really surprising also.
Do you think they used a different enzymes or?
But for this study,
Yes.
they also get it at 28. Also, as we
Yes.
they got the increase
Okay,
at
yes.
28, so it was similar.
Yeah.
But first we tried the higher temperatures because we thought that's where the literature says that's the optimal.
Yes, perfect.
And we expected more, but
Yes.
no.
No.
Yeah.
So I think we got through this one. I think you told us quite clearly that if you work with the fungi for a long period, you transform the organic matter into CO2, and that's, yeah, it is lost.
Yeah.
So that's why 7 days is. So it's, but yes, and that's my question here is that I could see when you're having carried out this survey, you have the mango seeds.
Yeah.
And what do you think will, if you look into a mango seed, I would assume seeds are not really easy to digest or to transform. Yeah. So what would you do if you have mango seeds? Because you have a lot of 100,000 tons of it.
Yeah, sure.
Again, I'm talking about your consultancy and working with this company.
Yeah, I think the mango seed outside it has a lot of fibers.
Yes.
But inside it has this soft part, which I think it's also full of protein or other nutrients. So probably you still need some fungi in case you want to degrade the upper part. And I'm not sure the content of the protein.
No.
So just by looking at it, it really needs fungi. But then yes. I think I have to test it to actually know how much protein. Or the easiest way is to actually just blend it with
yes.
another substrate. That's like the super easy one in case there's something available.
Yes.
That is less fibrous and see how they go.
So it's yes, but I thought there was a future in mango seeds because you showed it was such a huge amount of biomass you have there.
Yeah, it's there, and the company that I visited, they have a lot of it.
Yes.
And they can't use it because larvae don't grow actually.
No.
So they're also looking for the solution to that. Yeah.
Yes, and I think we have gone through all this. Phenols and bananas. It was interesting that that's a problem. And I think we have been through all this with ammonia. But I was really interested in this. I think maybe we have talked about this with blending in fish and banana peels because you have a lot of fish waste in Tanzania.
Yes.
Yeah. And so I think we also been through this. I'm sorry that you're seeing these slides, but that was the way I thought it was. What I was thinking of was you have this, you are saying 25% of fish waste is the right amount, and 75% is actually giving you a higher BCE, but it's not as predictable.
No, it's not.
Could it be because you're using this numerous you have a lot of species of fish? So could it be that it's not the same biomass you are adding, giving the larvae, to have you homogenized it so well? So that can contribute to your variation.
We did homogenize it really well. And because we based it on the amount of VS, we made sure we took the samples to actually check how much of the VS we're giving.
Yes, but also different species of fish.
But so the different species, I think, could be the problem. Maybe the composition itself. Because in this study, we did only the nutrition composition of the pure substrates at the beginning, but then we didn't do for every batch. We just say it will be the same. Yeah, maybe. So maybe it varied. That's why I think maybe the variation was big.
Yeah, I was just wondering because it was quite a
There's really
different
big,
fish, and
yeah.
as far as I know fish, there's was quite a lot of differences in the fat content in this, at least in the meat
Yeah.
of these.
Yeah.
Yeah, I think I asked you about this biomass conversion efficiency in itself, and I also think I've talked a lot with you, so let's just take a few of the questions. This one down here, I find it quite orange peels. Are they really the future for larvae production, like soda fly larvae production?
In one answer, if a standalone, no.
No.
But when diluted with other things, yes.
Yes. Even with all these limonene oils in it, and
I mean, if you dilute it, it's still it improves. So
yes.
if it's available and you have another waste, you can still use it. But alone, I think no.
Yeah.
It's not the future.
No. And we talk about these enzymes and phenols. It's also been biogas production. Phenols can be a big product. Probably fuel oils, it's called, one of them. And we talked about this availability of biomass, and I'm always coming back to the mango seeds because I thought there was a lot of it. Yeah. And then it's for the future. I just picked up these, and this is my last slide. So we come back to the big picture again. There needs to be a good economy in this. I think all engineers make these cost analysis and look at the market and all that. So where do you really see, if you're going to have a biomass, and it can be, so let's say brewery waste directly as a feed, but you can also produce like soda fly, and you have all the other wastes. So can you give me a few examples where you think that black soda fly is a better choice than any other treatments? Composting, for example, or direct use as a fertilizer, or it's maybe not a good question. Let's say organic waste from households is one of them.
Well, where can black soda fly be best?
Would would it be more cost efficient than just composting and use it as a fertilizer?
Compost and use as a fertilizer.
Yeah. Traditional composting.
Yeah. I think if the larvae are you get a higher value from the distal biomass. But in waste, for example, I think they complement each other and they work together sometimes in reality. And there's some type of waste which are probably good for the composting, like the really straw, like you said.
Yes.
Because they don't have sufficient protein, but then if you add something to it and then they are more valuable, probably it might be more valuable in the vegetal composting, because we get both the fertilizer that you get in the composting, and also we get the larvae themselves. Yeah.
Is this frass a better fertilizer in? Maybe I don't know. Have you worked with fertilizers or?
No.
No, so
but I know it's actually been applied.
Yes.
Yeah, and sometimes it needs a little bit more composting also, but you have already reduced the time for it, for the composting process in some cases. So, but then you just get still the protein, you put it, because the composting also doesn't take the protein. It doesn't utilize much of it, like to add value to it. No. All of it goes back, but this one you get both. Yeah. In both factions. So I think it's a really better. Yes. In terms of costs. Great. Yeah. So that's my last question. So
thank you very much for this discussion, and thank you very much for a nice presentation. And I already said your report was great to read.
Thank you.
So, and thank you for listening to us, all of you. And I think I leave the word to the chairman and the committee now.
Thank you for the nice questions.
Yeah, you're welcome.
For reading it.
Thanks a lot for that.
Yeah.
Then I will invite the evaluation committee. And actually, I might think that we start with the digital link if it is good. Susanne, can you, if you like, you can start.
Yeah, hello?
Now we can hear you, yes, please.
Yes, hello?
Yes, hello?
Yeah, okay. Can you hear me?
Yes.
Yes, we can.
Yeah, thank you so much for this this great opportunity. I think I'm very impressed with the cross-examiner for making my work easy. And I really want to thank Alice for this great piece of work that she has done. And having come up with such a concept of pre-treatment and substrate blending as a way of enhancing process efficiency in black soldier fly and composting is a very new concept, which is very, very important. I think and needs a little bit more research attention. So on a general note, I've read through the thesis, and I see that is actually well written. It's quite easy to understand, although in a couple of areas we find a couple of grammatical errors, which I think a second review over this will be able to address these issues. The research problem is actually very clear, and the objectives are quite smart and very straightforward. And the data that has been collected in this this work has actually answered most of the objectives that were set at the initial stage of this thesis. So the arrangement of thesis is also quite impressive, where you look at the introduction, the objective and the thesis structure, the background, methodology, and the discussion, and the half conclusion, and then recommendations. So, but what the and the final part, we see the papers that have been published have actually been put as the appendix. Also, an interesting part of the thesis is a concept where the citations of recent literatures from high impact factor journals have also been commended to be able to support the findings of this great piece of work that has been done by Alice. In overall, I think the logical flow in terms of the chapter on food industrial waste survey, which comes in at paper 5, which I also think Alice seems to have noticed along the line, would have, to me, be presented somewhere at the early stages of the thesis. I think around the first chapter of it, because it had to look at the inventory of the wastes that are available and making a choice as to which of them should be used. So I find it a little bit not properly placed if you send it as the last chapter of this thesis, because it actually highlights the status of waste generation, management, and quality of waste that was being generated. So in overall, the result of this chapter would need to justify why this waste treatment and blending is actually being carried out in this process. So far, when I look at the main achievement of this work and the significance to me, I find that you actually look at a large volume of bio waste which has been generated in the major food industry in Tanzania, which is very important because we need to look at possibility of how to manage this waste. She has actually highlighted a couple of waste streams like the banana peel, mango seeds, fish waste, and which are most available in terms of accessibility, the quantity that are available, the purchasing costs, and also the competitive use of some of these waste streams, which actually played a very major role in guiding her decision in terms of the waste that are being used for this study. And also I see that most waste needed to be mixed to be able to achieve a balanced nutrition for the black soldier fly production, except that the broilery waste and sunflower press cake were not part of this waste that were not actually being put to use. Also, the key aspect of pre-treatment methods which she used, like the biological, chemical, and the biochemical of banana and orange peels, and blending the fruit peels with fish waste, actually shows some level of improvement in terms of nutrient availability, and also increase the conversion efficiency of the black soldier fly larvae. Areas related to waste blending also have seemed to also increase or to minimize the effect of processing efficiency, which is also an interesting component of the work. Pre-treatment of the banana waste seemed to have reduced the bioconversion efficiency, and probably this could have been due to increased concentration of the tannin in the phenols, which she actually mentioned during her presentation. And so the microbial treatment of the peels also seems to have increased the amount of readable available nutrient and yielded some high bioconversion efficiency. Enzyme treatment at 28 degrees Celsius also seems to have played a very key role in also improving this whole process. So I'm quite impressed that most of this work has been published in very high impact factor journal, and 2 more of these are still under review. So therefore, to me, on a general note, the student works has a good mastery of the content of the work that has been presented in terms of design of the experiments, the data collected, and discussion of the findings that were obtained. Also, reading through the material method section, I seem that clear that at least has employed standard and methods and has produced sufficient data that answers the question, the research question that was set out at the beginning of the process. Some key aspect of, if you look at page 40, I'm still to understand, which will come to it later on, what some of the choices that informed the treatment periods that were actually being used as well, which we are going to address. So basically, Alice, I would like us probably to move down to some of the questions and comments that are related to the thesis after this brilliant outfit of your thesis, just for clarity, and okay, probably to see that we are actually on the same page, and actually get the thesis to state that the readers will be able to appreciate the great work that you have done. Mm. So from my end, when I look at the waste mapping that you did in Tanzania, I still find a little bit worrisome. Now, after these 3 years or 4 years of your work, what do you think is the current waste status of black soil production in Tanzania? Because I know there are a couple of companies that have been established, and from the waste stream that you focus on, none of this waste seems to feature top on the list in terms of mass production of this insect. You have Babo, you have Chanza, and many other emerging insect-based enterprises, but none of them none of this featuring the waste that you actually identified. So, what were the basis of you selecting this waste, given that we should be looking at a system where we could be developing this towards a business opportunity?
Thank you for your question. Well, my aim of this study was actually to identify what kind of waste that are available there. And then so I got the list of all companies, food companies under these 3 regions to say. And then I assessed all of them so that it can be known what kind of waste is there. And but this is only from food industry waste. We have other sources that I didn't look at, for example, market waste or household. So your question was, the way I understand you say the BSFL companies that have started, like the Bellevue, they're not using this type of waste, right?
Yes.
So what's the way forward? That's why we have tried to do this kind of pretreatments to see if it can actually make the substrates, like the wastes, more nutritious and available for the larvae. So maybe the first thing is to try and implement this on them, on the actual waste, and see if they work. Because why they are being repelled from using it is because the waste does not produce sufficient products like the biomass, and for them they're into business. They just want to make profit out of the system. So I think the first thing is actually implement it to actually see how to use the black sulfur to treat this waste and still increase the product from this waste, like the product value. That's where I'll start from.
Okay. Yeah. I still find it a little bit. Understand exactly why. Okay, if you choose those different type of waste, but the different method that you applied, as you said, for example, the pre-treatment of the pre-treatment method, the biological, the chemical, the biochemical, which and blending, you know, which if you look at it very closely, in terms of effectively, they do require very high capital investment. So it's a little bit difficult to understand how this should be cost effective if your intention is that these companies can depend on this waste stream. So how do you justify this? Because it doesn't seem to add up one on one if you have to look at the companies having to use this waste stream because of the cost involvement and the high quality technology that is involved in treating this waste. It doesn't seem to fit within the business model of any enterprise. Can you justify this?
Well, I don't know their business model, so I can't. But I think we have shown that if they're available, I don't know if they were mixing. They probably, I don't know what they've done, but at least they've shown that if it's there and you're mixing, you actually get a high conversion ratio, like it gets better. I don't know what they're getting now, but for example, that's an easy one if the waste streams are nearby, and also. But what are they using? Do you know what they're using now, for example?
So that okay, yeah, that's why I was trying to really get you to understand. So I'm good. The thing is, it means you've not done a very good background check because some of the study, as you said and concluded, it should have been feeding back into the system to improve the economy of the country and also to improve the livelihood of the people, or bring income to companies. The thing is, the reason I was asking this question is because the waste stream that you've used, getting accessibility to biological processes, getting ammonia is very expensive. Getting the enzymes is very expensive. Even availability is a big problem. Using this all the approaches, when you look at the ability to convert this waste to valuable resource that could be used to produce the insect and even to produce a fast fertilizer, in terms of the nutrient quality, it still remains extremely poor because from your thesis, because the protein levels itself is not up to standards of allowing the insect to grow effectively and to have the nutrient quality that is required for it to be a good marketable product. So that's why I'm asking now, if after biodegrading this waste using all this extensive and unaffordable approach, do you think this method you've used is cost effective at all? Or is just research for the sake of it?
I researched to see if they can improve the process efficiency itself, but I didn't really go further to do the assessment of it. I know that's why I mentioned in my presentation that this could have an effect. So there needs to be a preliminary study to actually check of these availability. It might not be for example, I've used the non-protein nitrogen. It could be another source of another type of nitrogen source that is available. So to check exactly what is available, because now we know in case we know the limiting factors in this type of waste that is there, and we know how we can actually make the nutrients more available now. But then we have to look into what's available locally that we can actually use to actually implement this and make it work out, I think, in the end. and then assess if it's like economical feasible before actually doing that.
Okay, I think just as Prof presented, trying to look at some basic concepts like some of the work. I think I made a mistake at the beginning not to have introduced myself a little bit. We talking about the fertilizer component. I'm looking at the work you've done and look at the aspect of us being able to enhance the larva biomass and the frass fertilizer quality,
yeah.
which is what your work should be guiding us to. And how can we explore this frass fertilizer as a novel fertilizer product in terms of the improving yield or in terms of nitrogen use efficiency? The reason I'm talking about nitrogen use efficiency because I'm driving towards some of the complexity of your work, and probably you might explain to us if this was due to the environmental setup of your experiments. We in the African context, we are looking at how can you recycle this in agro-industrial waste to produce this nutrient-rich organic fertilizer that could be more efficient in terms of the insect production. So that brings me back to your question of and which Prof actually narrated in his presentation. Where was this work carried out? Was it carried out in Tanzania or was it carried out in Sweden?
The survey in Sweden, the pretreatments and blending here in Sweden.
So now, based on the tools that you use in Sweden to establish these results, do you think it is affordable for our smallholder farmers? They will make up 80% of our agricultural population.
Well, I think the technology itself is flexible. You can do it using, I did it in a small scale in a lab in controlled environment, because here it's called different kind of environment. But at home, I think it can be done differently because one, we don't have this kind of weather. And in terms of doing like the process, you can use just what's available because it has been shown you can use different type of technologies using this. So I think it's still applicable, but not exactly how I did it, but in a context of that area.
If you are in Tanzania, how easy is it to get enzymes?
Now the enzyme part. The enzyme part, I think, is when it comes to the material now. Even here itself, it's been really expensive to actually buy it.
Yeah, that's what exactly.
And we couldn't even do more analysis that we wanted to do because it was really expensive. But we wanted just to check if it actually works. That's why in the end I said we have to check if we can either make the enzyme, and that's why we use just mixing of fungi, just normal fungi, to see, because we saw the enzyme works, and then let's try and see fungi, just mixing them to see if we can increase the enzyme activity. That would be easier and probably more applicable in that context with less cost. So I think it depends actually with the local context.
Okay, so the bottom line is that it is a very exciting research findings, but its applicability is still far-fetched from affordability to our smallholder farmers or to the Africa context.
I agree. For the farmers, it's really still very expensive.
That should be the starting point.
Yeah.
Okay, so we need to look at more affordable options.
Yeah, we need to look into that.
Okay, thank you so much. Prof also raised a very important question. Do you think what you have done, can you really call it a composting process, or it's just biodegradation of waste? Because you didn't answer that question. I know you played around it.
I think by the definition I have of composting that I've used, as I said, for my definition, it's still degradation of the organic compounds using the fly larvae and the microbial community.
Prof gave you a very good definition of composting in a dictionary approach, but it doesn't seem to be related to what you have done. Because I'm asking this because you see, he mentioned a lot of things in terms of stability, in terms of maturity of the fertilizer, and in terms of phytotoxicity, in terms of the hygiene, in terms of pathogens. Now, but your entire study seems just to have placed a black soldier fly on the waste, did all the manipulation you did with the chemicals and the approaches you did, and then when the larvae tend to pupae, you end and everything ends there. How sure are you, based on the elaborate presentation provided by Prof in his opening remarks, trying to give a a more broader picture of what compost is all about? How sure are you, or how confident can you say that what you've produced is a fertilizer at all?
I didn't really analyze the fertilizer because in this study I based it more on the DSL biomass, increasing it. But I know several studies have done it, and sometimes it needs a post-treatment to actually compost it even further with the normal composting conditions to actually make it now usable, like equivalent to the other type of fertilizers from the compost. So I can say I didn't really evaluate that because my work was just based on how to make the larvae obtain nutrients from the fruit waste, fiber-rich waste fruits, and I'd see if the pretreatment works. That was just mainly the concentration of this study. I didn't really go further into the fertilizer, that's why.
So what is the take-home message from your work? So what were you driving at? Just producing the larvae with poor quality and reducing the waste? Was that it? Because the entire presentation I see, it's gearing towards composting, and composting, it's about getting a fertilizer product. So now it means at the end of the day, there's no product at all from your work. Or what product have you produced from your work?
I have produced some bigger larvae, more biomass, by using pretreatments. I just didn't look at the other fraction of the process. I just only looked at one fraction.
Okay, that's fine. So I think now when I take you back to your experimental setup,
Yeah.
which was so much in a very small scale experimental approach, using just 200 to about 700 larvae, and yet the study talks about bioconversion performance. And we know there are a lot of studies out there that have clearly shown that density dependence is very, very important when it comes to biodegradation and bioconversion ratio. You know, there's a lot of errors that you get when you use very small size of very low density population of in a waste production system. So how do you justify this? And do you think this kind of small scale work that you've done, how practical is it to implement at large scale level?
Well, the aim was to actually evaluate if these pretreatment methods actually work, because most of them, when we began, they were not actually being tested before. That's why we saw doing it in a small scale was okay in terms of resources and time and just evaluation. But you're right, sometimes when you go into the industrial scale, you get different results because it's different larva density and probably just the logistics are different. And I think it's really good. But we have also tried some of these pretreatment and the fungi in a larger scale. So I think there's more need to look into this. That's one of the recommendations, to actually implement it in a larger scale. Now that we see it works, at least let's test it in a larger scale and see if it's applicable, if it's actually feasible to actually do it in a really larger scale. That's one of the recommendations of this study. Now that we, for the ones that are actually working.
So now, you say it actually worked. So what actually worked from your work? I'm still trying to understand the approach. As the first examiner was looking at the concept, you are the whole scenario where you were adding ammonia. Yeah. We are in a composting process, as he presented. We are looking at ways of preventing the ammonia, the breakdown of nitrogen to ammonia into the atmosphere. But you are adding the ammonia, and the insect rather needs the nitrogen for its own growth. How do you balance this up? Because what's the mechanism of you adding ammonia? Because the ammonia itself, adding it means it's already breaking up. It's going to get back to the atmosphere and evaporates, which could explain some of the reason you find you got your substrate drying up and so on. So how do you explain the use of ammonia when it has no role to play in terms of biodegradation or in terms of larva growth?
I think at least it helps in breaking down, but we don't have to use ammonia. Could use, for example, urea or other types, and in a larger scale, if the ammonia is not as available, like you say, because it is it's not easy to get, right?
No, I'm not talking about availability. I'm saying urea is still have the ammonia intact. We are talking about in a black sulfur production system. We are trying to avoid the breakdown of nitrogen to ammonia release, which now we go to the atmosphere and become a problem. Now you are instead adding the ammonia, which is already in a form that can easily vaporize. What is the concept of this? Because I don't see the role of ammonia in terms of biodegradation. I don't see the role of ammonia in terms of the larvae benefiting from the ammonia, because the larvae will benefit more from the nitrogen than from the ammonia that is found there. So what was the principle of you adding ammonia into this whole system?
The thing we thought it would also add some nitrogen and make more nitrogen available for the larva itself, for the growth. for the synthesis of ammonia, for the amino acids. That's why.
Do you have that in literature? Because I've tried reading on that. I can't find that. Because what I see people do is a way of preventing the nitrogen break stability to avoid breakdown of ammonia.
Because we saw, for example, for the ruminants, that's what they do. The ruminant animals, they add for them a nitrogen source, and then with the help of the microbes also, they use it for their growth in the animal production. So we tested that. And also in another literature, which all of them are in my thesis, we saw that addition of these ammonium, they help in the degradation of the fibers in the banana peels. And that's why I said, okay, let's do the testing and also in the fly larvae composting and see if it could also help. So there is some literature that we used, like the concept, to actually check if it can still be applied with the larvae treatment also.
That's a good idea. With the previous presentation, that Prof there was asking, the ammonia, is it the ammonia playing that role, or the ammonia is helping the microbes in the substrate to be more efficient in breaking down the fiber, or is it the ammonia? That's what I'm trying to find out. You need to get this picture straight, because it's not the ammonia. It is the organism using the ammonia for effective activity within the substrate. That's what I'm trying to get that picture out a little bit.
Yes,
okay. Okay, that's fine. Now, now if you look at your larvae, the growth, I see it taking 15 to 32 days as reported in your thesis. This is so much. It's a very, very long time, which is different from what has reported in previous studies. A lot of industrial production should be talking about 9 to 10 days of the larvae completing its developmental time in the substrate, but yours moving to up to 32 days. What do you think were the major factors that could have caused this kind of situation of the larvae taking much more longer time, and how can you improve on this? Because this is not, as I said, it worked, but in terms of large scale production, this cannot be a developmental process that will be more a business or any better profit margin. So what do you think? What do you think we can do to improve this?
Well, for the first study, we went for all the way to 30 days until the turn of pupae. But for these other studies, it varied, and that's because of the available nutrients again. Because what makes the larvae grow fast is how much they eat. So it's just a type of waste that we had. It's more fiber rich and less nitrogen. So it takes a longer time for them to actually accumulate the nutrients into their body and grow. So it takes a longer time. But if it was, for example, food waste, like it takes a shorter time of around 14 days. So I think it's just the composition. We have to improve the composition of the waste in general. And that's what we are trying to do to see if it could improve.
Okay, so that means you think based on the result you've generated, you need to combine the technology you have with other waste streams to improve the nutrient quality. Because
it could also do that. You could also combine with other better waste streams. You don't have to restrict yourself to only this. This is what was available. But in another context, maybe there's another type of waste that is even much better. So you can actually mix it. You can even mix with food waste if it's clean and safe. So I think there is no limitation on what you can mix the waste with.
Okay, now you talk about the pH, the low pH observed in the frass generated from the substrate, which was a direct implication because of the enzyme treatment. What is the implication of this low and high pH in terms of your experimental design? Looking at larva production and looking at the biomass conversion ratio. What do you think the enzymes did any good job, or enzyme can be left out from this whole process and you still have better results?
So you asked the pH, what effect it has, right, on enzyme? Oh, I didn't understand the question. You talked about pH and enzyme.
Yeah, the inclusion of the inclusion of enzyme in the substrate had issues with the pH levels.
Okay.
Either the high or the low. In case of high and low, what are the implications of these pH values in terms of the impact on your research findings?
Oh, well, the heated enzyme treatment and also other treatments that had low pH, it just means it was still the acids, because low pH means it's more of the acid. That means it was not a stable process, it didn't process. So these acids, they are the ones probably that hindered the growth of the larvae. And because also it means, because for the BCFL process, it's supposed to start from the acidic to alkaline, and even that's how it's structured. It just means that it affected the degradation process in one way of the unavailability probably, and some toxicity, the larvae. We don't know how it affects it. Maybe that's what you think. But it has been shown also, just in other literature, so low pH affects the final larva weight and the process efficiency. But then I don't know the mechanisms behind the low pH, exactly how it affects the larva growth. I'm just speculating.
Oh, okay, yeah, that has a lot of implication to the overall findings of your work, we all know that the ammonia has a negative impact on larva development. The enzymes has a negative impact on the larva development. So looking at page 61, what I see that the survival rates of the larvae was so bad. What was the exact survival rate of this larvae across the different treatment on average?
Page 61.
What was the range?
The survival rate of what treatment?
The larvae that were used in the different treatment. What was the range of survival?
For the table 6, right?
Page 61.
62 minus
61.
Oh, I have another book. Okay. So here, that's the one which has, here mine, I think maybe we have a different version. Mine is 62. Mine is 62 with the table.
Yes. So the low, you see there's a lower survival rate of the larvae. So what was the average survival rate of the larvae across the different treatments?
So we have different survival rates. We have the ones with high survival up to 97, and others have, like Rhizopa 7 days, it has 59, and then heat 66, and then ammonia and trichoderma 51, and those were the lowest.
Now the treated substrates, where you applied all this new technology, the survival rate was much lower compared to the untreated. So can you say the technology you applied works or it did not work?
I think for some, more than 85, I think it's good, but yeah, others were, it affected the survival. Others it was really low, like 50, the 60, they were really low, which you don't really want to see that.
Yeah, which is okay. That's but the survival rate is much better in a where there was not treatment on the untreated substrates. So that's where I'm trying to get a balance. But all the same, I think 50% upward is not really bad, but which means it can easily be adjusted. Then you were asking about.
I may you have a lot of interesting questions and things to discuss. We need some time for the rest of the evaluation committee too. So if you can limit yourself to one more question, some kind of general question or whatever question, it will be very good. Thanks.
Okay, thank you very much. So basically, with the work that you have done, what recommendations can you give either to entrepreneurs or to insect-based enterprises in developing countries like Tanzania, if you have to target scaling of this technology, and why?
The advice I can give, recommendation, is to actually apply it on all waste streams that are available and make it one of the options to actually consider, because it has potential to actually change the linear system into a circular one by producing products that's both the fertilizer and the essential biomass. So to prevent pollution, but also to recycle nutrients. So that's the advice, like to really start spreading this technology and actually work on it.
Yeah. So basically, from the work that you have done in overall, what are some of the pitfalls or the weaknesses you think you've identified from this work, and how can you improve them?
I think something that I would have done much better, maybe I would have loved to have considered, is to look into how for example, to determine how the cellulose and cellulose degradation happen and what type of carbohydrates, to actually understand the whole process better on how they work and the fungi itself, like the whole chemical and biological process into details. Yeah, I would have gone for that, so that you can know how to improve it much better.
So how can this work that you've done fit in the circular economy concept, given that is actually an incentive for adoption of this technology?
It fits because we are treating the waste and producing products that actually recycle. We're actually recycling, reusing the nutrients in what we are doing. So that's already is circularity, I think.
Okay.
Thanks a
Yeah.
lot. I think we have to say thanks to you at the moment, at least, because we need some more time for the rest of the evaluators here. And then I ask Maria who will go on.
Yeah.
You can use this one.
Okay, thanks a lot. I agree that your report has been really interesting to read, and I enjoyed this discussion today. And it's been a long day for you. I see you're tired, so I will try to keep short. But you already discussed the pH some, but that was something I also got very interested in because I was looking at your tables and your results. It seems to be very important, but you don't discuss it so much in your first introduction and so on, or discussion. But I see in paper 2 you discuss it more. Yeah. But maybe it's not just the pH of the substrate that it's important because looking at, I try to compare the survival rate and the weight of the larvae, because it seems when it's successful, the pH goes up to above 7 or so. Yeah. So, I mean, and you know that the pH hinders the growth of the larvae. Yeah. So, did you think of measuring the pH during growth? I mean, maybe there were something that quite rapidly increased the pH in the substrate, and then the larvae could grow for a longer time period at optimum pH. Or maybe was this something you were thinking of?
For the first study, I measured the pH throughout and like every week. But for these 2 studies, I only measured at the beginning when I'm giving them the food, and at the end when the residue, the pH residue. And no, I didn't really follow it up in the experiment, like to know how it's changing. Not really, no. Yeah.
And I mean, because when I was thinking about this, it can be quite interesting to actually think about the larvae feeding pattern. Mm-hmm. What do they prefer to eat? Is it the microorganisms or is it the organic matter, would you say?
No, they like the organic matter.
It's the organic.
Yeah, I think they might just help in the degradation. Yeah, I mean it's part of them also, but it's the organic matter that actually make them grow.
Okay, so because so if you have air present and you have microorganisms there, the acids will be quite rapidly degraded, but microorganisms is quite an easy substrate microorganism. So I'm thinking, because when you heat your substrate, you don't get so good results.
No,
right?
We don't.
Yeah. So I'm thinking, because in all organic material you have natural occurring microorganisms that can consume the acids maybe if you put it in the air. So is it the risk that you actually kill off your natural occurring microorganisms by heating?
Maybe, but even at lower temperature, I'm not sure if, because some microorganisms in the 60, most of them don't survive that. Probably.
Depends on how long.
Yeah, because it was just 24 hours for the lower temperature. So I'm not sure how much they are deactivated. Yeah.
But you also use the heating for...
Because it was just until 50. I mean 50. For the 120, I think it really deactivates.
But you're thinking about the enzyme, when you add enzymes, right? I'm thinking about when you, for example, you heat it up to 120 degrees.
Yeah,
and
that I think you really deactivate completely. Yeah.
Okay, so maybe because you were discussing the production of phenols and tannins, that maybe can also be that you actually also kill microorganisms that be good and help them to increase the pH or help the degradation of the organic material.
Probably, but yeah. But then let me check 1 minute. For this other study, I want to check the pH actually. What was when we just heated without the enzyme? I want to check the pH.
Yeah, I looked at the pH.
For the biological.
So for all experiments that worked,
Yeah.
the pH increased.
Yeah, because even for the ones which was 50 degrees centigrade, it was still a little bit lower and without enzyme, biological enzyme. And then there is that control with no enzyme. It was still like the pH was also low, but I'm not sure if for that we also deactivated all the microorganisms with the 50 degrees centigrade.
Did you kill them all?
Yeah.
Hard to say.
Yeah.
Some of them can survive.
I think some of them can still
Yeah,
survive at
yeah.
that temperature.
Yeah.
That's why I'm not sure if it's just the. I think it's really part of it, but I'm not sure if it's the only reason. That's my point. Yeah.
Yeah, because I checked the pH and I saw, was it in paper 3, I think, where you did this PCA with all the parameters.
Yeah.
It was really nice.
Yeah.
But then you looked at the initial pH and also the end pH. But
Yeah.
I think it's important to actually look at the pH during growth as well, because it might really impact on the growth of them, or like. It seems like it, at least. You had one exception, and that was with the orange peels. The peels with the enzyme 0 days. I mean, then you actually had a low pH at the end, and both at start and end. But except for that, the pH seems to be very important. I would say.
Yeah, maybe that's a good idea to actually do it like that to see if it can. But then, what would you do in case you saw it decreasing? Maybe you understand just the process.
That was actually what I was thinking of, because it seems like maybe going too high is not good either. I mean, some of them were around 9.9. That's super high. So I was thinking about some buffer capacity. So substrate that could maybe, of course, animal manure is perfect in regard of capacity, but that's maybe not the substrate you would like to use.
Okay.
So, but having something that will actually, neutralize the pH, I think that.
Yeah, that could be something.
And then I was also thinking about the enzyme pretreatment that in paper 2.
Yeah.
This is on page 19, and you already discussed this a little bit, but I just maybe I interpret your discussion wrong. So I just wanted to check with you, because you say that having this enzyme mixture and you heat do you mean that you think you get that the reading here, hemicellulase activity, that change and you get other products? Or I'm just thinking how you discuss the impact by heating at 50 degrees instead of having at 28 degrees. So you were saying that maybe other carbohydrates are formed. I just want to, because increasing the temperature would of course increase the activity
of the enzyme.
Yes, of the enzyme. So, but when I read this, I read a little bit interpret that the enzymes will give other products.
No, I mean, we meant increasing the activity of the enzyme, and if we increase the activity, it means they degrade more, fibers like the cellulose into available carbohydrates. That's what I meant. Yeah.
So more of the fibers are degraded, but because you discuss that they might have formed arabinose or galactose, so there are other products formed.
I mean, it's still, because in the degradation process, it is the long chains which are being broken through the interaction of the enzyme and the active sites of the the micromolecule and then it breaks it into other forms, like simpler chains, right? And then, so the point was, instead of getting mesosyl glucose as the end product, you just get another polysaccharide in the middle that is more available. It doesn't go all the way into carbohydrates. So that's the enzyme does its work, but then afterwards.
So you mean they just do half of the work, so they...
It's the kind of activity that is there. What do they do? Yeah, because they break it, but then afterwards, what happens? Yeah.
Yeah, I was just thinking because I would maybe not say that the products are changed. Because,
no. They don't give another type of product because enzymes are very specific.
Yeah, exactly.
Like protease works on the other. But then afterwards now, like maybe one product hinders the other one, like the function, or maybe they're denatured. Like that's what. There's something that hinders the whole process with the heat. Maybe they're denatured in one way, or maybe there's some product that when it's more and it hinders the other one, and so I don't know what really happens, but I think there is some kind of hindrance in the enzyme activity, but it's not like they produce another type, but I just think the process is hindered in such a way it doesn't go all the way into producing available carbon. Probably. I'm not even sure. Yeah, it was just a speculation.
Okay. Yeah. I would recommend to think read this discussion part again before you submit the paper, because it's easy to interpret it wrong. So, and also and also one small comment. Because sometimes you write that the fiber content increased, but maybe for example, if you have shorter or longer incubation time with the microorganisms, and so maybe it's better to write less efficiently degraded or something like that. So I think I should give the word to the next. Thanks a lot.
Thank you.
Okay, thank you really very much. It was really nice reading this thesis. I appreciated it very, very much. I appreciated pretty much every aspect of it because I also like the organization. I thought that by organizing it in that way with this, let's call it a preliminary part in the laboratory, and then the part related to what are the substrates available in the country where I live, I suppose, and where I would like to work, so that okay, let's prepare and then use it. So I really like also that type of aspects. Pretty much many, and I also agree with the colleagues about all the other aspects. Really, I enjoyed it. The reading was really nice, and so on. And many of the questions have already been addressed, because pretty much if you are the last, you can expect that something has already been in the mind of also the other components of the commission. However, there are still some aspects. And one, for example, is really a curiosity. I understood the reason why you choose banana peels, because in the end it is also something that is in Tanzania. But I didn't understand why you choose also orange peels. Maybe they are part also of the Tanzanian agriculture. I ignore that, but I was kind of, okay, why? Also because we know from the literature that orange peels, they're rich in these phenols, in these essential oils that we know already that they could be detrimental to the growth of the black soldier fly. So yeah, my question is, why the orange peels?
One, it's a lot of streams of orange peels worldwide, and including Tanzania and here. But at the same time, we wanted to see, because also in other processes, it's a problem still. So we wanted to see how the larvae would actually grow on them also. Yeah.
Yeah, yeah. And also if the type of treatment that you choose were able somehow to
twist it, yeah.
Yeah, and then you had the surprises.
Yeah,
that
again.
yeah, yeah. So on page 43, when you talk about the methods, you say, and also when I checked the articles, you said that you use this average temperature for the rearing of the black soldier fly larvae, 28.28 plus minus 0.8. So according to the literature on the black soldier fly, usually it is around 27. So I was wondering why you decided to have a kind of higher temperature, and also related to the conditions. Here it is not written, and also I checked in the articles. I was wondering about the relative humidity, because you know that is crucial. So I really was just, to be sure about the conditions that you used.
Yeah, the literature, it differs. Actually, it varies. Some use
I know.
7, others 29. It varies. But here, I use the one that I use because it's being used in this big production that has already been established for a longer time, and that has been shown to be optimal, like optimized. So I just went with that.
And the relative humidity, what is
it's
it?
Around 75, 60, I'm not sure. Yes.
Okay. Yeah, pretty much. That one is according to the literature.
I didn't really measure it every time. So.
Yeah.
It's around there.
Then,
but it's also being controlled.
Yeah. And then a question which is more like, let's say, practical. So if you were to transfer this technology, as I think you are supposed to do in Tanzania, so according to both your experience and according also to the literature, and considering that probably the best thing that you want to do is to obtain larvae which are very rich in proteins, because that's what everybody wants if you want to use these larvae for feed for animals. So what would be really the best combination of substrates and or pretreatments? So really your aim is to maximize the protein content in the BSFL larvae, and so what would you prefer, considering especially your country, according to what you know? And
can I please repeat the question? Sorry.
The aim is to maximize the protein content in your larvae.
Yes.
So what substrates, especially considering Tanzania, and eventually and or pretreatments, would you use?
The best substrate, because it's blue waste, which is, according to my study, right, is not really available. But that's like unsandbar. Those are the best in terms of nutrition composition. But then when we go in terms of availability and usual, I think I would still go for maybe banana peel and fish waste. That is available. Both of them are really good because none of them works alone. Or banana peel, like combination of the substrate, but for the case of Dar es Salaam, we don't have much of the sunflower just yet. But then if you're in that region, maybe you can go for that or something else.
So you need some more preliminary research.
Yeah, you need some. It depends with where you are actually.
Yeah.
Yeah, because for example, in Dodoma we don't even have fish waste. That means you have to actually look into what can work with Sven papers. Yeah.
Then reading your article on number 3, in the discussion, I was really negatively surprised about the amount of heavy metals in the fishes of the Baltic Sea. I said, oh my goodness, I was really concerned. So because for your experiments, you used basically the fish offals. So yeah, I mean, it's really the discarded part. I was wondering, okay, is that comparable or similar to what you would find considering the fish offal? Because you didn't really use the fish, you used what the discarded part, yeah. So the gut, this type of things. Do you think that would be comparable? I wish not to the fish you find in your country.
Yeah.
Yeah, I mean, in terms of especially these dangerous stuff, the heavy metals.
I think you still get the inside the same, but then the composition, I'm not sure if it will be the same with the heavy metals and yeah, maybe we'll have to.
Yeah, I hope it's not the same.
No, I hope
I wish it's not the same, because that was really terrible. And related to the, okay, you performed experiments here in Sweden, and here we are in Europe.
Yeah.
So you probably know that here in Europe, we are not allowed to use these animal stuff for rearing anything, you know? So I was wondering, and this is a European regulation, I was wondering if you have the same problems in your country or not. Because here it prevents a lot the development of the technology, because unless you are able to use something which is, a waste, it is an animal waste, then okay, you're stuck. You cannot move forward. But maybe that in your country you can use it. And so,
for example, in my country, there is no yet regulation on using this kind of waste on BSc for technology. I don't know now if it's, but there have not been policies and about it. So you can still use them, but I think it's also still very good to check, because these prions or contaminants, they are always good to check, because it involves feeding people with these products. So I think it's very, very good to check still, to make sure that they are not there.
Yeah. Final, it's just curiosity, or because you realized that you and everybody that has reared the black soldier fly, we realized that really this is not the best insect to valorize animal byproducts, because about the content of fat, these type of things. So if you would improve the technology considering this type of target, so animal byproducts, whatever comes out, fish offal or slaughterhouse waste, these type of things, because the black soldier fly is not really the best, what would be your solution to utilize also that type of substrates? Animal origin substrates, waste, animal origin waste.
But those ones can also be used directly as animal feeds.
Excuse me?
They can also be used directly to the animal feeds, actually, most of the animal products.
Okay.
They can be used directly.
Yeah.
Yeah. That's also one option.
I mean,
or you could use to other type of insects.
Exactly.
Yeah. That type of insects.
Because I'm an entomologist and I'm kind of all over the world we have so many species of insects. Why only focusing on one?
Yeah, there are also other types of insects that you can actually use.
Yeah, and in Africa, where you have the different the highest biodiversity.
Exactly.
Like, okay, come on, let's use also some other species.
Yeah.
Thank you very much. It was really, really nice.
Thank you. Thank you all.
Insights. Åke, do you have anything Yes.
I can
Yes.
say it. Thank you. Let's see. It's on, yes? So yeah, thank you very much, Alice. I also enjoyed reading your thesis and your presentation. I have quite followed you in your work, and I think that I see a very clear progression in your work, and I think it's ended up in a very nice thesis as well.
Thank you.
And as even though being this reserve, it's maybe expected to at least ask one question, and I will not prolong this very much more. But I will make maybe a little bit follow-up on the enzyme which Maria raised. I was quite fascinated by this very quick response on the 0-day addition. And you also explained that you were not able really to analyze the different, you measured the crude fibers, for example.
Yeah, we did.
And then in the suggestions or the recommendation for future studies, you also say that you would like to explore other types of enzyme cocktails.
Yes.
And yes, we all know that enzymes are expensive, but let's forget that for a while and say that you, from a more scientific point of view, would like to reveal more knowledge about what kind of enzyme activities is needed for this fiber fraction in order to facilitate or increase the growth of the larva. Do you have any thoughts about what enzymes or enzymes combinations you would like to test?
One of the enzymes I think we should test, can be tested, would be inclusion of pectinase, because especially the oranges have a lot of it. And yeah, that could be one of them. Even the bananas have pectin, so maybe that could be one of them.
Because I was just thinking when reading, and maybe I misunderstood a little bit, but it seems like from what you're writing on page 78 that these pentoses, which originate from hemicellulose, seems to be a little bit problematic, and it's much better than with the glucose coming from the cellulose.
Yeah.
So would one option be to just try with cellulases and glucosidase and avoid the hemicellulases, or is there any other kind of issue which you will not cover then?
Yeah, because the cellulose, as we showed, maybe there might be some hindrance, but still we have some other like pectins. That's why I think we can. But then removing it completely, I mean, they were already the cellulose and glucanase, they were there, and they actin specific. So I'm not sure remaining the two of them could improve further, unless this actually hindered, if it is true, it hindered, maybe. So maybe it's what should be examined, maybe. Yeah, I'm not sure, because I don't know what really happened to hinder the process.
And first also to know how much hemicellulose and how much cellulose is in the banana peels, for example.
Yeah.
Okay, thank you very much.
Thanks a lot. Then, are there any questions from the audience?
Nothing from online.
No technical questions? No IRF questions? Well, if not, then I think it's time to say thank you to Alice and to Sven and to the evaluation committee, both here and in Nairobi. And I will by that soon close the session. But first, we will say that there will be a closed meeting at the upper floor at the department of our department, 5th floor here. And then there are also some drinks served, I think, outside. And then we will wait for the decision of the evaluation committee. And if we can start the meeting 4:40, it's very good. And yes, and Dr. Tanga, you have to connect to that meeting, but I think that you are informed on how to do it. And with that, I think now it's time to close this session. Thanks a lot.